So we'll go to Senator Rennick.
Okay, hi guys, how are you going? My first question, there are now over 118,000 adverse events recorded against the COVID vaccines. That's an injury rate greater than 1 in 200, and that's basically more than all vaccines put together since 1971. What is it going to take for the TGA to take these injuries seriously and suspend the vaccine rollout until the risks of the vaccine are reduced to a much lower tolerance level? And what is the point of having a database of recording all these injuries if you're not going to use it as a signalling system?
Senator, the purpose of the database is to receive reports to actually feed into identification of whether there are signals, safety signals. The reason why we have received many more reports in the DANE database for COVID vaccines is twofold. Firstly, there has never been a vaccination program in which Australians, the overwhelming majority, in the 90%, have received 2 doses, and as we've heard earlier...
most people get their scheduled vaccines as children.
Senator, if I may answer, the majority of Australians are not under 5 years old. So if you look at the absolute number of individuals...
But over the last 50 years,
Senator, if I can complete my answer. If you look at the absolute number of Australians who have been vaccinated in this program, it dwarfs previous vaccination campaigns. The second thing is the awareness...
But the rate. I asked about the rate, and that's what I want to get to. It is a very high injury rate.
Senator, if I can answer your question, I do not accept your assertion it was a very high injury rate. I'm talking about the rate of reports. And as we've explained before, there's a very big difference between someone putting a report, and in many of the health systems, it is a mandatory for doctors to put in a report even if they don't believe there's an association, if it's related in time. So
okay, thanks. Can I ask the next question now? I've got
I am conscious that Senator Rennick's got quite a few questions, so if we can just have...
Sorry?
Senator Rennick's got quite a few questions and we've got limited time, so if we can try and just have some...
Okay, sorry, I've just got to get through these,
sure.
So it's got to be a short answer. So I noticed in your recent half-year report that over 50% of the reports are actually from the state governments themselves. Mm-hmm. And what do you do to vet those reports and make sure that the state governments aren't vetting reports? And I'll just put in there that I know a cardiologist who's contacted me. He lodged 3 adverse event reports for myocarditis, and the Queensland State Government went back to him and said he can't do that. It has to be the GP. Now, why is it that cardiologists...
He can report them directly to us, Senator. There's absolutely
well, no, because he's been told that Queensland Health...
He can report them. You can report them.
Well, I'll pass that on.
A doctor can report directly to us.
Okay, so Queensland Health are telling specialists that they have to go through Queensland Health. They can't lodge them directly with the TGA.
I haven't heard of that.
Well, I have. I've heard it firsthand.
Well, they can report directly to us.
Okay,
I'll
pass that on.
Any individual in this country can report an adverse event to us.
Okay, thank you, because I think it's important that you realise that some of these state health departments are vetting out adverse event reports. Okay, just letting you know. Okay, so a TGA whistleblower has given me data this week that says that the number of reports of death on the DANE database is actually 2,142. When the TGA does its weekly report, why does it only report 810 deaths as received and reviewed, rather than give the total number of reports received?
So the total number of reports received is a couple of % higher than that 800, and we checked firstly for duplicates.
Well, that's not what I've been given. I've been given 2,142.
Well, that's not what I've been given,
right.
Senator.
Okay.
So we checked for duplicates and we checked for fraudulent reports.
Right.
Very sadly, there are some quite malicious people who are making up death reports.
Right.
So, for example, we have one report in the name of a doctor called Dr. Fake Doctor.
Right.
We have
so how many? Okay, so I accept that. How many of them are?
Less than 2%,
right,
we believe, are removed from being duplicate reports or clearly fraudulent reports.
Yep.
And even in those cases, there may be a doctor called Fake Doctor. We follow it up to see if there is such a person and whether it is a genuine report.
Okay, so they're only 2% of fake.
Or duplicates.
Yeah, okay, that's fine. We just need to quantify that. Thank you. Given Pfizer has tried to cover up releasing all of its COVID vaccine trial data for up to 55/75 years, has the TGA asked Pfizer for the reasons why it refuses to be transparent with the trial data? And is the TGA concerned about the fact that Pfizer withholds data given its prior criminal convictions?
We have the trial data that was required under the conditions under which we and other global regulators review vaccines. We have the complete trial data from Pfizer. I think you're referring to the subject of a US Freedom of Information action. That is very different to the regulatory data package. The regulatory data package for these vaccines can be up to over 200,000 pages of data, so it is extremely extensive.
So I'll note though that that data was based on vaccines produced in the laboratory and not vaccines commercially produced, and that matters because there's a different manufacturing process. Has the TGA and I know that you signed off based on trial data where they used the laboratory.
Not true,
vaccine.
Senator. No, that's incorrect, Senator.
So you basically signed off, when you signed off in December 2020, you signed off on data, vaccine trial data that use vaccines commercially produced.
So the vaccines that are used have to be made using a process that is either the commercial process or directly equivalent to the commercial process. And one of the reasons, for example, that we took a long time to review the Novavax vaccine is we went back many times to the company until we were convinced that the small-scale production was equivalent to the commercial production.
So what date did
that's
Pfizer
an absolutely
give you?
critical part of the process.
Okay, so what date did Pfizer give you the data based on the commercial production of the vaccine and not the laboratory production of the vaccine?
So we have the data from the clinical trials from Pfizer, and we have the data for the commercial production. I'll also remind you that every single batch...
So what date is that?
Sorry, if I can finish my sentence, Senator. I'll remind you that every single batch of vaccine is going through TJ laboratory batch release.
Okay, and how much variance have you got in mRNA?
Every single batch of a Pfizer vaccine has met the specifications.
And what's the variance between intact RNA and degraded mRNA?
I can take the question on degraded RNA on notice, but it is extremely low.
Right, okay.
But we do assess for that and we do test for that and we reject a batch if it did not meet the specification. So what is your cut-off
rejection batch?
I'll take that specification on notice.
Okay. So I refer to Freedom of Information number 3604 where the TGA was unable to provide documentation on genotoxicity issues related to the provisional approval of the Pfizer COVID vaccine. Has the TGA assessed the genotoxicity risks of the vaccine given a large number of modifications to the spike protein, including the replacement of amino acids to speed up the production of spike proteins, the addition of adenosine nucleic acids to prolong the life of the mRNA, and the replacement of uridine with surouridine, which is used as a signal by the ribosome to stop the production of the protein? Given these significant modifications to the genome of the virus spike protein, shouldn't there have been significant quality control and testing around the potential risks of the modifications?
So, Senator, those issues have been looked at some length, including the use of pseudouridine.
So why weren't they released as a part of the Freedom of Information request then?
Because that information is considered as commercial in confidence.
Well, that's not what you said in the Freedom of Information request.
As you realise, our regulatory submissions do contain commercial in confidence information.
Well, that might be commercial in confidence, but that's going into people's bodies, and it's very important that people are aware that the spike protein that they're receiving from the vaccine is not the same genomic sequence as the spike protein from the virus. And I think people, as per the immunisation handbook, have a right to know what the risks and benefits are of the vaccine.
And that's why you have a medicines regulator who publishes an Australian safety report. So, for example, the AusPARS, the public assessment report, was published the day that the Pfizer vaccine, the very day, at the same time the Pfizer vaccine was approved.
Okay, and I still have a right as a senator, and as a senator, as a representative of the Queensland and Australian people, I've got a right to ask for that information, and I want to know all the data on the genotoxicity of the modified mRNA vaccine, because people think that they're getting a spike protein that's equivalent to the virus, and they're not. It's been supercharged to produce spike proteins, and I think the public deserve to know that.
It's been modified to produce a stable product which will produce a good immune response.
That stays in the body longer, that increases its half-life.
There is no evidence for long-term persistence of the spike protein.
Okay. Just because there's no evidence doesn't mean to say that there isn't evidence of no dangers. There's a big difference where you put the no. Okay, there's no evidence because there hasn't been enough genotoxicity studies done in the human body.
I disagree with your statement, Senator. I believe that the studies have been adequate.
Senator Rennick.
Well, that's what we asked for in the Freedom of Information, and it wasn't
Senator Rennick, can I just grab
forthcoming.
your attention for a moment? You've got less than 3 minutes to go, and you also had questions, I think, for the Office of Gene Technology and ATAGI. Did you have that or not?
Sorry?
You've got less than 3 minutes to
Okay, no,
wrap up,
I'll
so-
stick to this one, but I'm happy for the Department of Health to answer this one.
I'd like that information too, Senator Rennick.
Yeah, hang on. Yeah, okay. So I asked last year and I'll ask again, has there been studies on the safety risks of multiple booster shots to the immune system of 4, 5 or 6 shots within a short time frame of 2 years?
There have not been studies in a regulatory submission of, say, 6 shots. There have been real-world studies of the safety and efficacy, say, of a 4th shot of vaccine.
Real-world studies, so no actual trial data.
No,
prior
they are
to-
studies, a real-world study where you have many thousands of people vaccinated for a 4th time, followed for an extensive period in the health system is...
But was there any studies done
published in
prior
an
to that?
international journal.
I'm
is
aware of these
quite
real-world trials because people are being used to work this out, but I'm asking, did you do any trials for 4 and have you done any trials for 5?
The regulators do not run clinical trials, Senator.
Okay, well that's just, okay.
No regulator in the world runs clinical trials.
Has the manufacturer of these drug companies or the drug companies run trials on 4 or 5 boost?
Well, the manufacturer was
doses?
closely involved, but with these real-world studies, because that is how you gain data. So if you're working with a population that's already vaccinated, the way to gather data on the safety and performance of a vaccine is to use people who have had, say, 3 vaccinations and are having their 4th vaccination. But in this case, they are closely monitored for any possible adverse event and also
So you're
closely
using
monitored for efficacy.
real people outside a trial setting, and is this an active or passive surveillance?
Well, in this case, it becomes actively surveyed if it's for a research publication in a top-shelf journal where the study is to say, what is the rate of adverse events after a 4th dose? Is it worse? Are there new adverse events? They are actively followed, and then a study is published in a top-shelf international journal.
Authorised G. Rennick, LNP Chermside.